The process of spermiogenesis consists in the conversion of round spermatids into sperm cells. During this process, which takes place while the spermatids are still attached to each other by the cytoplasmatic bridge, the spermatids modify their shape by elongating the nucleus, rearranging internal organs to create an acrosome over the nucleus, reorganizing the position of mitochondria in a mid-piece, which will serve as the engine to activate the developing tail in order to swim, and discarding most of the cytoplasm. The entire process can be divided in 4 phases. The first phase, called the Golgi phase, consists in the initial elongation of the cell. The Golgi apparatus produces enzymes which are incorporated into a single acrosomal vesicle which gets located at one of the poles of the nucleus. At the same time, the nucleus condenses, leaving inside a tight package of DNA in the form of chromatin. In the second phase, the cap phase, the acrosomal vesicle covers about 2/3 of the nucleus, adding a very thin double membrane layer of enzymes over this area. At the opposite end of the nucleus, a centriole is placed to serve as the initiation point for what will be the axonome or tail of the sperm cell. In the third phase, the acrosomal phase, the elongation process continues and mitochondria start migrating and organizing into a thick mid-piece, and the tail continues to grow. The cytoplasm starts to be displaced towards the tail end of the cell, leaving the acrosome as the external part of the new cell. In the last phase, the maturation phase, the elongation is completed and the tail is fully grown. Here, most of the cytoplasm and other organelles are reabsorbed by the Sertoli cells, leaving a small cytoplasmatic droplet attached to the mid-piece of what is now the new sperm cell.